Hello everyone. So in this session today, we will talk about enzymes and there will be three lectures on enzymes alone. And basically, we are still talking about proteins because almost all enzymes are composed of proteins. So let's have a quick revision on the functions of proteins. First of all, it plays structural roles such as forming keratin in your fingernails or collagen inside your tendons and ligaments. They also form contractile motor proteins such as myosin that is bound to actin. So you have learned with me in cell biology that these are cytoskeletal proteins. And then proteins also can have transport function such as hemoglobin that transport oxygen, while glucose transporter they will transport glucose molecules across the plasma membrane. And you do have proteins that comes with defensive functions such as antibodies, proteins with storage functions such as myoglobin that store oxygen and ferritin that stores iron. And you also have proteins with regulatory functions such as insulin and growth hormones. And last but not least, you have proteins that play catalytic function, which are enzymes. So that is the focus of this session today. If you look at the human genome, roughly a quarter of our genes encode for enzymes. So that is a significant portion of human proteins, and that is a huge number. So why do we need so many enzymes in our body? Well, this is the answer. So this picture shows you the metabolic pathway in our bodies. You can see that there are many dots which are different substrates. So for each dot to be converted into another dot, our bodies will need enzymes to catalyze these biochemical reactions. And because enzyme action is so specific, each reaction is catalyzed by a specific enzyme. So this explains why we will need so many enzymes to carry out the metabolism. And showing here is only the summary of our metabolism, and there are many more pathways that are outside of this diagram. So the history of enzyme study can trace back to about 300 years ago when people discovered that secretions from the stomach can be used to digest meat. And then in 1800s, people used saliva and plant extracts to convert starch into sugar. You can also try this yourself by chewing your rice long enough. You will turn starch into maltose, which is a disaccharide, and that will give you the sweet taste of rice. And then in 1850s, Louis Pasteur, who was a renowned microbiologist, coined the term ferment, which are enzymes that turn sugar into alcohol during fermentation. But he thought that ferments are some sort of inseparable forces from the living yeast cells. So this view is called vitalism, in which living things have a vital force that is inseparable. But his idea was overturned by Buchner because this is what Buchner found in his experiment. Fermentation was promoted by molecules that continue to function when removed from cells. So these are enzyme molecules, and they are not a type of force, but rather they are solid molecules. And eventually, Frederick Kuhn called these molecules as enzymes. Well, the journey didn't stop here. In 1926, Sumner isolated the crystal of urease, which is a type of enzyme that break down urea, and he postulated that all enzymes are proteins. And then Northrop and Kunis furthered the study by crystallizing even more enzymes such as pepsin, trypsin, and other types of digestive enzymes. And it turned out that all enzymes are made of proteins. And it is not until 1950s only we have a scientific hypothesis on the mechanism of enzymes. So John Haldane wrote in his book Enzymes by saying that weak bonding interactions between an enzyme and its substrate might be used to catalyze a reaction. And we know that it is true today. So in the late 20th century, due to the use of X-ray crystallography, NMR, and other techniques, the structure and mechanism of many other enzymes have been elucidated. So what are enzymes? Well, first of all, they are made of proteins, which are biological macromolecules. But you do have exception as well. So some enzymes are actually made of RNA, so we call them RNA enzymes, but they are very, very rare. Well, all enzymes are catalysts, which means they speed up chemical reaction and they show specificity. So one enzyme normally will catalyze only one type of reaction, but again, they are exception as well. And enzymes are sensitive to changes such as physical changes and chemical changes within the environment, and their activity can be controlled by the organism or by the external intervention. So we'll come back to this and cover everything about enzyme regulation in the subsequent lectures. So as mentioned just now, enzymes are made of proteins predominantly, with the exception of a small group of RNA or DNA enzymes, but essentially you can assume that all enzymes are proteins. So one key thing about enzyme activity is that they are totally dependent on the structural integrity of the native protein conformation. In simple terms, enzyme activity is depending on their three-dimensional structure. So when an enzyme has lost its original folding due to high temperature or due to the presence of other organic solvent, then they will lose their activity as well. And if you look at the sizes of the enzymes, it can be very diverse. It can be as small as 12,000 daltons, or it can be as big as 1 million daltons. So some enzymes can function as a single unit, or some enzyme will come in the form of multi-enzyme complex. So it is a very diverse group. And then last but not least, some enzymes require no chemical groups for activity, which means they can function as a single unit, or some enzymes will require the presence of additional chemical component, such as a cofactor. There can be any type of inorganic ions, or the presence of a coenzyme, which is organic molecule, or some sort of metallo-organic molecules. Okay, showing here are some examples of cofactors for enzymes. So you have something like copper, ferric ions, potassium ions, and so on. So they are all required by certain enzymes to function properly. So as a result, it is always better to drink mineral water rather than a reverse osmosis water, which has no metal ion. As you can see, our body will require a range of different metal ions so that these enzymes can function properly. Now, just one more thing. Some heavy metal ions can be toxic because they can actually replace the metal ions cofactors within the enzyme, such as mercury and cadmium, which make the enzyme non-functional. Okay, aside from cofactors, some enzymes will also require coenzymes to function properly, and these are organic molecules. And these coenzymes actually serve as transient carriers or temporal carriers of specific atoms or functional groups during enzyme catalysis process. So you can see that from this table, the precursors of many core enzymes are actually vitamins, especially from vitamin B family. You have B12, B2, B6, B1, and so on. Okay, here you have some terminology. First of all, holoenzyme is an active enzyme because it is bound with its coenzyme, while an apoenzyme is non-active because it is not bound with its coenzyme, and therefore a holoenzyme is an apoenzyme plus its coenzyme. Okay, next, enzymes are classified by the reactions that they catalyze. But how do we name the enzymes? Well, it's pretty simple. We can add the suffix ase or ase to their substrates. For example, urease is an enzyme that breaks down urea, while protease is the enzyme that breaks down protein. Now, how about pepsin? What does it break down? Is it Pepsi? No, definitely no, right? So before the specific reaction catalyzed was known by the scientists, they can use any name. But as scientists have learned more about enzymes, as more enzymes have been discovered, now the naming of the enzyme is governed by a professional body known as the Nomenclature Committee of IUPNB, so that you do not have the freedom anymore to make fanciful names. So for example, ATP glucose phosphotransferase is the enzyme that will transfer a phosphate group to a glucose molecule. So it's very standardized and very clear. So that is the benefit of this new nomenclature, that you can predict or you can tell the function of the enzyme just by looking at its name. Okay, here is a quick question. So what do these molecules have in common? Over here, you have a list of drugs like Tamiflu, Relenza, aspirin, and so on. And over your right hand side, you have a list of poisons such as sarin gas, alpha amanitin, and so on. Okay, the answer is they are all enzyme inhibitors, which are molecules that can stop enzyme from functioning. Okay, this is just a joke, but you don't want to eat mushroom that contains alpha amanitin, as they are enzyme inhibitors that can inhibit RNA polymerase from functioning. Okay, as mentioned just now, Tamiflu and Relenza, these are anti-influenza drugs that function as enzyme inhibitors. But which enzyme do they inhibit? Well, they are actually enzyme inhibitors for neuraminidase. So what is neuraminidase? It is an enzyme that is found on the surface of flu virus. So these are your neuraminidase, the one in green and red. So what these enzymes do is that they will remove neuramic acid from the surface of host cells so that the virus can escape the cell. As you can see, when there are inhibitors inside the cell, so this neuraminidase activity is being inhibited and there will be no viral release. Okay, showing here is the 3D structure of the neuraminidase protein, and the red patch here is where the Tamiflu will bind. However, a mutation at this site can lead to Tamiflu resistance. For example, a replacement of lysine into arginine. So this explains why a flu pandemic can still happen in modern days because viruses they have the ability to mutate all these protein and they render the old vaccines or the old drugs useless. Okay, here you have a second example of enzyme inhibitors that can function as a drug, which in this case penicillin, which was discovered back in World War II and it has saved many lives from infection. So what it does is to bind to an enzyme known as transpeptidase, or sometimes we call it a penicillin binding protein that is required in the synthesis of bacterial cell wall. So with the presence of penicillin, the bacteria will lose the ability to synthesize cell wall, and that eventually leads to cell death. But bacteria will not be defeated so easily because they can develop antibiotic resistance. And in this case, it is called a penicillin resistance. So what gives penicillin its effect is this structure, what we call a four-membered beta-lactam ring that will bind to transpeptidase and inhibit the enzyme. So this gives the ability of penicillin to kill the bacteria. But somehow, some bacteria become resistant to penicillin by producing beta-lactamase, which is an enzyme that can digest away this beta-lactam ring structure. So these resistant strain of bacteria are known as superbugs. And the good news is we can actually inhibit this beta-lactamase by feeding them with potassium clavulanate, which is another type of enzyme inhibitor. And the real challenge with this antibiotic resistance lies in the presence of these multi-drug resistant organisms. And we have very limited choice of drugs that can fight against these MDROs. So as a future biomedical scientist, this is an important issue that you need to understand and try to solve. Okay, one last example of enzyme inhibitor acting as drug is aspirin. So many senior citizens are taking aspirin on a daily basis. So it is your homework now to find out more about aspirin. So which enzyme does it inhibit? What is the mode of action? And what are the other functions of aspirin? So apparently aspirin has multiple functions. So please go back and look for the answers.